it will be mechanobiology with optical tweezers. And I'm using the abbreviation OT for optical tweezers. Well, in this first lecture, I will discuss what they are, the optical tweezers, and how they work. Then, how you use them to extract from a cell membrane what are called membrane tethers. And this has important applications to determine the parameters, elastic parameters of the cell membrane. And I'll explain the connection, which is given by what is called the Helfrich-Kahnum theory, and discuss the application to neurons. In tomorrow's lecture, the general subject is cell membrane nanotubes, which again will have an abbreviation, CMN. I'll discuss what they are, and then what is their functions, or what are their functions. We'll see that they have a very important role in the propagation of infections, as well as in cancer transmission. The last lecture will be a seminar. It's based on a paper I just submitted, and the title is Origins of the Nervous System of Eukaryotes and of Life. Okay, now for a general introduction, and by the way, I'm probably not quite the youngest physicist in the audience. So a long time ago, when I was a young physicist, I would be able to lecture and to address people in the back of the auditorium and hear their questions and so forth. But since I'm slightly older now, I have trouble hearing, so I'll need an assistant to take the microphone to whoever is asking the question. Thank you. And please speak slowly and loud, okay? But I welcome questions at any time. Now, it used to be said that the 20th century was the century of physics, and that the present one would be the century of biology. Well, my own case is a good illustration of this point. From about the last half of the past century, I was a theoretical physicist working in scattering theory and in quantum optics. Just the year 2000, I underwent a kind of phase transition to experimental cell biology. What was my motivation? It was the invention of a very remarkable new instrument, which is optical tweezers, which revolutionized cell biology. It allows to manipulate living cells without damaging them. It can detect very tiny forces of the order of piconewtons, which are typical of the cellular scale. So it's very adapted to studying cell biology. It also detects displacements of the order of nanometers. in a limiting situation, you can use it to study how a single single molecule interacts with the cell. And this has led to a new area in biology, which is called single molecule biophysics. There is a journal with that name. I will not be discussing single molecule biophysics here, but it has also led to a new domain in biology, which is very active, which is mechanobiology, which is what I will be discussing. And it has a large number of applications. One of them, which was mentioned in yesterday's colloquium in statistical mechanics, you can use optical tweezers to sort of experimental realization of a Maxwell demon. And one of the most important applications was to follow the motion of a motor protein within the cell. For instance, also to follow step by step the transcription of a DNA molecule. Okay? So you can see how it steps, measure forces, and so forth. Now, because of these very extraordinary features, I decided to establish an optical tweezers laboratory at the Federal University of Rio de Janeiro. It's called the COPEA Lab. COPEA is for Coordination for Programs of Advanced Study. And besides applications to biology, I already mentioned statistical physics. There have also been applications to fundamental physics. And an example is our project that we have currently on, which is to study the long-range behavior of Casimir forces, which has never been detected and requires measuring forces at the level of femtonewtons. Okay. Any questions so far? All right. Now let me begin with this explanation and discussion of what are optical tweezers. Okay, optical tweezers are optical traps for neutral particles which are typically of micrometer size. For instance, which is the main thing used, transparent microspheres. Now, we are all familiar with the fact that radiation transmits energy, and you can feel this on your skin if you go to Copacabana Beach during the summer almost at any time. And you can have a demonstration how it is affected by focusing. If you focus the sunbeam well enough, you can use it to burn a piece of paper, okay, and also to molest people, which should not be done. All right. Now, besides transmitting energy, radiation exerts forces. It produces what are called radiation pressure. And radiation pressure pushes an object, and the basic proposal was made long ago by Johannes Kepler to use this to explain why a comet orbiting the Earth, this is a very elongated elliptical orbit, when it is close to the Earth, the tail of the comet points away, okay, like this, from it. And Kepler ascribed this to the time they had the corpuscular theory of light, so that a shower of light particles is pushing on the comet tail, which is made up of essentially ice particles. Now, besides pushing, the radiation can also pull an object using what is called the gradient force. What is the gradient force? Well, again, when I was a young physicist making a lecture demonstration on what is called the power of sharp points, I used to rub a comb on my hair. At that time, I had some, and show that it attracts small pieces of paper. You can do this very easily if you have hair and a comb. Well, how does this happen? Here is the explanation. If you have a sharp point, at the point of a comb, which is electrified, okay, say with positive charge, then if you have a neutral particle, the field is much stronger near the point than far away. Well, this positive charge will polarize a molecule, for instance, and it will attract negative charges and repel positive ones. However, the negative charge is closer to the point where the field is stronger, while the other positive charge, which is repelled, is further away. So there is a kind of tug of force here, and the end result is that this dipole is attracted to that sharp point. So that's the gradient force. And as with the transmission of energy, the stronger the focus, the stronger the gradient force, and the more strongly can it pull particles. Okay? Well, that's the tug of force that I describe here. And for that to be strong, you require highly focused laser beam. Now, if you are fans of Star Trek, you know about the tractor beams. Here is the big Star Trek thing. It's using a tractor beam to attract an enemy rocket or something, okay? It's the same thing as an optical tweezer, but the optical tweezer operates at the microscopic scale. All right. Okay. On the left, you see what is the typical setup of an optical tweezer. Well, okay. In fact, you can demonstrate an optical tweezer at a very, even undergraduate laboratory level, if you just have a lens and a laser. You can attract the particles and show just with that tool. However, if you want to apply this to cell biology, you use a microscope which allows you to do many more things at the same time. So what do you require? Here is a laser. And the laser is usually in the infrared, typically a neodymium YAG laser. Why is that? Because it's a transparency window for water, and the cell interior is mostly water. So it will not be absorbed and heat up the cell and do damage. That's why it can deal with living cells. Okay, now here it goes through a telescope, and then here you have a dichroic mirror. Why? Well, you want to deflect this to the objective of the microscope, which is the lens that will do the focusing. And at the same time, you want to see what is going on. So you also have visible light illumination that goes through, because this is dichroic, to a CCD camera, which is monitored, and you can follow this in real time on the screen, okay? Now, the kind of Bible of optical tweezers is this book by Arthur Ashkan, which is called Trapping of Neutral Particles Using Lasers. It has several hundred pages, and it's beautifully written. It's a collection of papers following the whole development of optical tweezers and their applications, and each paper has comments by Ashkan. Okay, now here is Arthur Ashkan, who invented optical tweezers. Okay, Ashkan worked at Bell Labs at the time that Bell Labs supported what is called blue sky research, which is research based purely on curiosity without immediate applications, industrial and so forth. Unfortunately, well, that led to several Nobel Prizes won by people working at Bell Labs. But nowadays, it has changed. It wants to have applications and motivate what is done by applications, and that means it's no longer getting Nobel Prize kind work. Okay, now Ashkan worked at Bell Labs for several years. He is a very shy person and very difficult to get in touch with. And he really deserved 3 Nobel Prizes. The first one for doing the first demonstration that you can trap atoms with laser beams using the gradient force. And this work he did with a postdoc called Stephen Chu, who later won also a Nobel Prize. Chu was following my undergraduate classes at the University of Rochester. And, well, he was not an A student. He was B+. But he told me that it was fair, that the work he did not deserve an A. All right. Now, the second Nobel Prize that Ashkan deserved was that he invented laser cooling. And laser cooling is what allowed to produce temperatures very close to absolute zero. And that did yield a Nobel Prize, but not to Ashkan. It gave the prize to Steve Chu, Claude Cointrenucci, and Bill Phillips. And then the third Nobel Prize that he deserved was for the invention of optical tweezers. As I said, a revolution in cell biology. Okay? Well, he got it very late. He was in his 90s, and here is his picture. He was the Nobel Prize in 2018. I got to know Ashkan when he was investigating. He was levitating microspheres, which were just oil droplets, and following fluctuations in radiation pressure, which were extremely sharp resonances. And he found out that at the time, working with me theory, I had a paper in which it was shown that you could detect these resonances with precision comparable to quantum electrodynamical precision. So he was very interested. He wanted me to help identifying these sharp resonances, and he wrote to me. Now, later, I met him by attending a colloquium where he was, very soon after the invention, demonstrating how you can use optical tweezers to trap bacteria. And he had a video in which he showed a whole set of bacteria trapped at the focus of an optical tweezers, and you can see them trying very hard to escape. Then he switched off the beam, and the bacteria go away in all directions. Well, I was very impressed with that, and therefore I decided to build this optical tweezers laboratory at the Federal University of Rio de Janeiro. Okay. Now, how do you apply optical tweezers? Well, first, the orders of magnitude for their features. As I already said, you use a neodymium YAG laser typically, that's 1,064 microns, 1.64 microns, sorry, because it is a transparency window for the water in the cytoplasm. Now, what is the force range? Well, typically, less than 1 piconewton to hundreds of piconewtons on a transparent microsphere, micron-sized object. And as I said, this is not the lower limit. We're detecting femtonewton forces as well. Okay. Why do you use these microspheres? You use them as force transducers. Why? Because you can compute the force on a microsphere by using diffraction theory, and precisely what you use is me theory, which is one of my specialties. And so you can compute the force on the sphere due to the optical tweezers. Now, the sphere is attached to whatever object you want to attract. And this could be a bacteria or something. And here is the focus beam shown, and here is the focus. And for very small displacements, the object which is attracted is here. For small displacements, you have Hooke's law. So if you measure the displacement, you measure the force. Okay? That's how you know what the force is being applied by the microsphere on the object. All right. Here is the Hooke's law. The force is the elastic constant, which is characteristic of the beam multiplied by the displacement. How is the force exerted? Well, if you have a ping-pong ball and you drop it on a hard surface, it bounces up. So the hard surface transmits momentum to the ping-pong ball. And by Newton's second law, it exerts a force on the ball. But by Newton's third law, the ball also exerts an equal and opposite force on the table, and that's the force that gives rise to radiation pressure. Okay? Now, here is how the gradient force makes this microsphere be attracted. What happens is that if you take the rays that the laser beam has as at its boundaries, take an example here. Well, that ray is deflected by refraction at the surface as it hits the sphere. Then by refraction, it travels to the opposite side. It is again refracted. So it starts with a momentum P1 and ends with P prime 1, P prime 2. Oh, I'm sorry, I showed the wrong one. This is the ray on this side. It's reflected. It's refracted here and refracted out again. And so it changes its momentum from P1 to P1 prime. That gives rise to a transmitted momentum delta P1. On the other side, you have the same thing, and delta P2. And if you take the opposites of both using Newton's third law, the net transfer of momentum is delta P, which points upward. It points, so it attracts the sphere to the focus. Is that clear? Any questions? Please ask. All right. Now, in order that you can compute the force, you need to determine the spring constant of the beam, which is called here kappa. Well, I'm using this notation later for a different thing, but okay. So how do you calibrate? Well, you can do it experimentally by comparing the trapping force with the viscous drag force. If you have the microsphere traveling through a fluid, and whatever you do with cells, they are immersed in a fluid chamber, then by viscosity, it will have a viscous force, and you can equally well in the microscope, you move the chamber past the sphere, and then you measure the escape velocity when the sphere is no longer able to stay in the beam and gets out. And then you use the viscosity law, which is essentially the Stokes law, that the force is proportional to the velocity, and the proportionality force is 6 pi eta times the velocity, where eta is the viscosity. Okay. Now, this is an experimental calibration. Another experimental calibration is to follow Brownian oscillations of the trapped microsphere around equilibrium and study the power spectrum of the Brownian fluctuations. And both methods are well described in this paper by Neumann and Bloch in Review of Scientific Instruments. However, you can try to get an absolute calibration from first principles if you know enough about how to compute the force from first principles. And what do you need for that? Okay. As I mentioned, it needs Mies theory, and it needs to use a representation for the focused laser beam, which was first suggested not for lasers, but for ordinary light by Peter Debye, and Emil Wolf extended this to a focused laser beam. Okay, by using these ideas in our lab, we finally, after several years, managed to produce a first principles theory for absolute calibration. This was published in 2012, and here is the experimental test. It requires taking into account that the beam undergoes several effects of deviation from geometrical propagation, in other words, aberrations, and there's spherical aberration and so forth. So you have to determine and take into account all the aberrations. And when you do that, you get what we called the Debye spherical aberration, MDSA plus theory, and this differs from the original theory without aberrations, which is shown here in dashed line, and the one finally including aberrations is the full line, and here are experiments, and you see that it agrees with experiment without any fitting parameters. So you can absolutely calibrate the optical tweezers. Okay. Now let me start the applications to cell biology. So explain membrane nanotubes and begin by showing what you have seen several times in by previous lecturers, the idea of how the cell membrane is structured. Well, here you see, using different how do you call it, dyes, a picture of a cell in which in blue is the cell nucleus, and in green are what's called microtubules, and finally in red the actin filaments, which are very close to the outer cell membrane. Here is a picture of actin filaments. These are both polymers and biopolymers. For the actin filaments, they're like, well, a kind of necklace with pearls or something, twisted, and the repeat distance is about 25 nanometers. Now, microtubules, which are capable of much stronger forces, have this kind of structure, which you might compare with corn, you know, and the typical repeat distance is, again, of the same order, and it's also the radius, okay? All right. Now, here is the structure, typically, of the cell membrane. You see that below is inside the cell and outside is here up. And you see that it is made out of these sorry. Well, the phospho thing, which have, main thing is they have hydrophobic region and hydrophilic region. So the hydrophilic region likes to be inside the cell where there is plenty of water, and the hydrophobic one is outside. So you have several attachments outside, typically carbohydrates, glycoproteins, and you have, okay, you have here proteins, typical alpha helix one. Okay, you have cholesterol, and here are, that's very important, the region below here, as I showed above, there is a kind of cushion made out of the actin filaments, and that plays an important role also. You have to take into account. Okay. All right. Now, here is an application of the optical tweezer for tether pulling. Tether is how you, like a leash that you use with a dog, okay? That's a tether. So here is the cell lying on a surface. It has this typically elongated shape. And here is the microsphere attached here. And what is shown, this double cone, is the laser beam. So what happens is that you can displace the laser beam to pull on the cell membrane by using the gradient force. And what happens is that it pulls out a nanotube, in other words, a cylindrical membrane tube, which is very narrow, and that's what is called the tether. And you see here that you have, if you know this displacement from equilibrium, you compute the force which is exerted by the tweezers, and at the same time you know the displacement. Okay. Now, what are the elastic constants of a cell membrane? There are two elastic constants. First, because it is a phospholipid bilayer, it will resist bending. Why? Because by bending a membrane, here is illustration below, you are expanding the average spacing of phospholipids on one side, you're compressing them on the other side. And that's the reason why you get the curvature. Okay. Now, what is the relationship? How do you define the elastic constant for bending? Well, if you have, instead of bending, just a straight line stretching, you have Hooke's law. The displacement is proportional to the force, and you get the elastic constant. But there's a similar generalized Hooke's law if you are bending something like a rod. Here you have a straight rod to begin with, but you exert a torque on it. You apply forces on both sides, and you can bend it. And what happens is that it becomes curved, and you measure the curvature, which is here defined by the constant C. And the generalization of Hooke's law is that the torque is the bending modulus multiplied by the curvature. And the curvature for just a straight rod is just the inverse of the radius of curvature. And then the constant kappa here is called the bending modulus. And just with the linear Hooke's law, which tells you that the energy associated with this is 1/2 of the elastic constant multiplied by the square of the displacement, the associated potential energy involves 1/2 of kappa, the bending modulus, multiplied by the square of the curvature. And a line integral of this over the length of the whole rod, I'm still talking in one dimension, gives you the potential energy. For a membrane, which is two-dimensional, it is characterized by two radii of curvature, or principal radii of curvature, R1 and R2, and you define the mean curvature as 1/2 of the sum of the principal curvatures. And the associated free energy is the surface integral of 1/2 of the bending modulus over twice the mean curvature squared, which is now extended over the whole area of the membrane. So it's a surface integral. And of course, you also have surface tension, and that is the usual thing you do also, which is well known. So for instance, in a balloon, you have the surface tension. And in tether pulling, you really have a tug of force, a competition in free energies of the curvature and of the area which is associated with the surface tension. Okay? In other words, you have a tug of force. Okay. As I mentioned, a cylindrical nanotube extracted from a cell membrane is called a tether, membrane tether. And suppose it has a length L and the radius R extracted by an optical tweezer, pulling force F at equilibrium. And if you want to find out what happens, you have to minimize the free energy, subject to the boundary conditions, experimental constraints, which introduces Lagrange multipliers. So the total free energy is the sum of three contributions: the free energy associated with bending, where it's 1/2 of the bending modulus times the mean curvature squared, integrated over the area of the bent membrane. Then there is the free energy associated with increasing the area of the membrane by an amount A, and this is sigma times A, where sigma is the surface tension. And finally, since you're performing work by pushing on or pulling on the membrane, you are to pull a tether of length L, the free energy is minus F times L. So if you add these three contributions, you get the total free energy that must be minimized. That's exactly what is done in the Helfrich-Kahneman theory. If you want to do it as an exercise, you can do the calculation, and I will discuss the solution in tomorrow's lecture. Any questions? No, there should be. Okay. Yes, but I need someone with a microphone.
Just a quick question. So, how does that the nanotube or nano wire gets attached to the sphere?
How you attach the sphere, the microsphere?
Oh, yes.
Yes. Okay. In some kinds of experiments, you use a kind of glue that biologically is used for precisely this kind of purpose. However, in our lab, we don't do that. If you just touch the cell with the microsphere, it gets attached by reorganizing the actin inside. That's much easier. No more questions? Okay, then till tomorrow. Thank you.